And so what is this major histocompatibility complex anyway? It's really just another form of communication. It's a communication receptor. We talked about cytokines kind of assisting with communication, and the MHC complex is going to be very classically associated with binding to these T cell receptors, and usually the MHC receptor is either going to be on kind of like your everyday nucleated cell or an antigen presenting cell. Now, when we're talking about class 1 MHC, we're talking about the kind of the standard MHC receptor. That's how I remember it. Class 1, it's kind of just like your baseline MHC receptor. It's pretty much going to be on almost all of your cells, particularly most nucleated cells. So remember, when we say nucleated cells, we're not talking about red blood cells, right? Red blood cells will not have MHC1 because they're not nucleated. Okay, so this has repercussions down the road when we talk about some of the pathology, but that's just something to keep in mind. Now, the key is the MHC1 receptor is essentially looking at every cell, and it's making sure that there's kind of nothing suspicious going on with all of our cells. So it allows for recognition of healthy versus infected cells. So you can imagine, right? So if you just have some normal cell here, okay, so this normal cell is doing what it's supposed to be doing, and on its surface here, it has this kind of MHC1 receptor. And, you know, if I have some T cell nearby, okay, so I have some T cell over here, this T cell will also have a T cell receptor. And this, you know, kind of healthy cell might present some of the self-antigens. So there's a lot of proteins and that type of thing that are in the cytosol of the cell, and some of those proteins might end up going into the rough endoplasmic reticulum and eventually getting transported to the surface of this MHC1 receptor. And this T cell might recognize that and say, hey, okay, that's a self-antigen. And remember, we already talked about this. In negative selection, we had our T cells undergo negative selection, right, in the thymus when they were maturing. And in negative selection, we tried to bind to self-antigens, but the T cells that survived did not bind to self-antigens. So this T cell that's then seeing this healthy cell putting a self-antigen here, it's not really going to bind to it that tightly. And so there's nothing to worry about, right? Now, let's see what happens, though, if I had a virus come around. So if I had some virus come around, okay, so here's some virus. All right, and this virus then gets into this cell. So it's going to go into the cell, it's going to hijack the cell's machinery so it can make more viral proteins. Well, the cell, what it will do is it will take some of these proteins, like we just talked about, it'll bring them into the rough endoplasmic reticulum, okay, and then it'll break them down and then transport them to the surface of the cell. And so now when this T cell's kind of jumping around here, checking out all the cells, it's going to see, hey, look, there's an antigen that's presented here that isn't really supposed to be here. It's going to bind to this antigen because it's a foreign antigen. It's going to be the right antigen to match that specific T cell receptor. And then we'll talk about what this T cell does, but you have to keep this in mind, right? On all of these kind of normal everyday cells, they're going to have MHC1 receptors, right? They're going to have MHC1 receptors. So that means that this cell that's binding to MHC1, what did we say? Well, it must be CD8 positive according to the rule of eights, which means that this must be a cytotoxic T cell, cytotoxic killer T cell. Okay, and so you'll see what we'll talk about in the next video. This cytotoxic T cell will then essentially destroy this cell using granzyme and using enzymes to essentially punch big holes into the cell membrane, because the idea is this cell, we're basically sacrificing it because we don't want this virus to then kind of burst that cell and then have even more viral particles infecting other cells. So that's the concept. And remember, what this cell will also do, it will also release some interferon alpha and interferon beta to the nearby cells. Okay, and so it'll essentially try to tell them to upregulate their MHC using interferon alpha and interferon beta. Remember, this will then upregulate MHC1 on the nearby cells so that there's a higher probability that if those other cells are infected, they will be detected early by more cytotoxic T cells. MHC is not just in humans, okay? So the way that we kind of classify it or grade these, we call these the human leukocyte antigens. And it's just important for you to understand that for MHC class 1, you can see there's only one letter after this human leukocyte antigen or HLA class. So the specific one here, A, B, C, there's only one letter, okay? So one letter for MHC class 1. You'll see in MHC class 2, there's 2 letters. So that's how you remember it. One letter, think MHC class 1, 2 letters, think class 2. Now, sometimes you'll get questions on the actual structure of the MHC receptor. We can make a whole 2-hour video on, the different receptors, but just make sure you understand that these receptors have beta 2 microglobulin. Okay, so that's this component here. And you can see that the MHC1 receptor is kind of asymmetric compared to the MHC2 receptor. The MHC2 receptor looks a little bit neater. It looks a little bit more symmetric. So sometimes you'll see that, this MHC1 receptor, they'll say it's either asymmetric or has 2 polypeptides of different lengths. So just kind of keep that in mind. There's a little bit of asymmetry here, and it also uses beta 2 microglobulin. Now, the MHC1 receptor complex, like I was talking about here, it's developed in the rough endoplasmic reticulum, which is a little bit different than the MHC class 2. So again, these are just kind of some of the details that sometimes come up that you want to be familiar with. And the key thing that we talked about here is that, again, a virus has to usually infect a cell, right? So it's usually an intracellular pathogen, something that infects the cell so that then we can, once it's in the cell, we can take its proteins, put it in the rough endoplasmic reticulum, eventually get it through the Golgi back up to the MHC1 transporter. So again, this is going to be, MHC1 is going to be usually intracellular pathogens for that reason. Okay, and again, CD8 positive T cells are the ones that are going to recognize MHC1. We talked about that already, usually leading to cell-mediated destruction of that cell. Now, the particularly high yield thing, this is very high yield. If there's an absence of an MHC1 receptor, in other words, if the T cell comes by and says, hey, this guy doesn't have an MHC1 receptor, if there's a cell that doesn't have MHC1, natural killer cells will tend to destroy those cells. Okay, so natural killer cells are kind of on the hunt to make sure everybody's got their MHC1 receptor. And you might be saying, well, what's the point of that? Well, remember, if you have a virus that comes by and infects a cell, through evolutionary processes, these viruses have learned to downregulate MHC1 receptors, right? Again, we're releasing interferon alpha and beta because we want more MHC1 receptors, but the viruses have trying to downregulate them. Everybody's trying to kind of compete to see if these cells have an infection, okay? And the viruses obviously don't want to be detected, okay? So they can downregulate MHC1. The same thing is true for cancer cells. Cancer cells can also do this. Natural killer cells are always on the look for an absence of MHC1. And again, MHC1 receptors will be upregulated by interferon alpha and beta, and I think we talked about that earlier. Now, when we're talking about MHC class 2, we're not talking about just your kind of like everyday nucleated cell. We're talking specifically about antigen-presenting cells. Remember, we said this is going to be macrophages, dendritic cells, B cells, right? These are all going to be very classic for presenting antigens to either B cells or T cells. And again, the HLAs are going to be 2-letter HLAs, specifically for MHC class 2. Now, again, we talked about the receptor has chains of equal length, which is going to be different from the receptor in MHC class 1, which is going to be asymmetric and have beta 2 microglobulin. The other thing that sometimes comes up here when we're talking about MHC class 2 is this invariant chain. When we talked about MHC class 1, we're talking about an intracellular pathogen. For MHC class 2, because we're talking about antigen presenting cells like this macrophage here, this macrophage, right, how is this process going to start? This macrophage is eventually going to get into some contact with some bacteria here. Okay, so let's say that this is a bacteria. Okay, so this macrophage is going to get into contact with this bacteria. It might attempt to destroy it or phagocytose it. And once it does that, it's going to break that bacteria up in the phagosome, right? And eventually it'll fuse with a lysosome to break down that intracellular bacteria. Okay, so it's going to kind of keep them in these phagolysosomes to break down the bacterial contents. So that's what the macrophage is initially going to do. And we already kind of talked about how this process originates. Now, once you get this bacteria in this phagolysosome, you're going to acidify it, right? Macrophages, we also talked a little bit about the superoxide radical formation, respiratory burst pathway. So you're essentially breaking this bacteria down. Now, once you break it down, this macrophage is going to take components of that bacteria and present it on MHC2. Okay, and so once you do that, now you can have a CD4 positive T cell come around, bind to that MHC2 receptor, detect it, right? And then it can activate this macrophage. And it does that through interferon gamma classically. Now, notice what's different here. When we talked about the first pathway, we talked about this whole thing happening because it was an intracellular pathogen that infected the cell, that being the virus. Here, it was an extracellular pathogen that a macrophage digested and then presented to MHC2. Now, in the process of this happening, there's kind of like a little chain on the MHC2 receptor inside of this phagolysosome, and that's this invariant chain. And the invariant chain just essentially prevents the antigen from binding prematurely. And it's one of those things that, I wouldn't remember every step of the process unless you have a lot of extra room in your brain, but the invariant chain is one component that I would associate with MHC class 2. So think of it like a little hook on this MHC receptor as it's in this phagolysosome before it gets transported to the cell surface that essentially prevents the antigen from binding before it's broken down completely. Okay, so that's just a simple kind of way to remember that. And remember, when we're talking about MHC class 2, extracellular pathogens, CD4 positive T cells, usually resulting in a humoral response, at least primarily a humoral response. Whereas when we're talking about MHC class 1, we're talking about intracellular pathogens like viruses in particular that result in CD8 positive T cell mediated destruction, usually more of a cell mediated process.